Hello, everyone. I'm Hunter Swilling, a sophomore here at the University of Wyoming studying physiology and molecular biology. I currently work in Dr. Yoon Lee's lab, and we study circuit mechanisms of neurodegenerative diseases by using imaging technology from fluorescence microscopy. So let's get a little bit of background about neurodegenerative disorders. Neurodegenerative diseases are characterized by the gradual deterioration of the structure and function of either the central or the peripheral nervous system. There are a lot of examples of this disease, and it's a pretty widespread factor in a lot of patients' ultimate death. In fact, Alzheimer's is the 9th leading cause of death in America today. The other examples of neurodegenerative diseases include Parkinson's, Huntington's, amyotrophic lateral sclerosis, and frontotemporal dementia. All of these are characterized by that slow, gradual decline in neurostructure and function. Over there on the right-hand side of the screen, you can see the normal brain tissue compared to someone who suffers from mild cognitive impairment. You can see that gradual deterioration in both the central and the peripheral parts of that brain. And then on the right-hand side, you can see someone suffering from late-stage Alzheimer's disease, in which large parts of the brain have simply ceased to function, and the remaining parts are significantly decreased in their ability to function and their general abilities. Age is a significant risk factor for neurodegenerative disorders. In fact, the average age of onset of a neurodegenerative disorder is about 60 to 70 years of age in humans. The neurobiology of these diseases is still not completely understood, especially as it relates to neuronal activity in the brain and the frontal temporal complex, which is where a significant amount of the functioning of the brain comes from. That is what my lab aims to study, that neuronal activity, and how it is impacted by various neurodegenerative disorders. So in order to study neuron activity, first we have to visualize it. And this is accomplished using some pretty breaking ground technology in a pretty clever way. So our lab uses a mouse model. It's a genetically modified mouse that's modified via viral injection to either express or show certain traits of neurodegenerative disorders. The mouse is additionally oftentimes an older mouse, and old in a mouse is only about a year old. So it's actually not that hard to get an older mouse. So it shows both of those things, those risk factors for age and those neurodegenerative disorders. We use Grin lens and miniscope imaging technology to be able to determine and see the neuron activity in the mouse's brain. A Grin lens stands for a gradient index and allows us to visualize and refract light to be able to look at neuron activity in the mouse's brain. That miniscope is a tiny mini camera literally mounted onto the mouse's head. It's important that it's mounted and small because it allows for the mouse to engage in free movement while having the miniscope still implanted on its head. This utilizes calcium imaging technology and calcium markers to literally see neurons as they fire and as they are activated. It's pretty great breaking ground technology, and it was only perfected in the last 2 or 3 years. So I'm really lucky to be able to have such an impact and see such a cool lab. So after the miniscope has been implanted in the mouse via surgery, and after the mouse is all healed up from that, we perform behavioral tests on these mice. We do this for both control mice and mice suffering from neurodegenerative disorders. We have a variety of tests, such as social tests, which is when a mouse is exposed to another mouse, and we see the neuron activity in there as that occurs. We have novel object recognition tests, which tests memory in mice by first exposing them to an object, and then waiting a certain amount of time and re-exposing them to 2 different objects. One object that they've already been exposed to, and then a completely new object. In mice who have a functioning memory system, we expect them to spend significantly more time in that novel object rather than the familiar object. But if they spend about equal amounts of time in both, it's indicative that their memory is significantly impaired, as is common in many neurodegenerative disorders. We also utilize a radial arm maze test, which is the test that I'm on, and I will elaborate on further in this lecture. So we use this miniscope imaging technology to allow us to visualize neuron activity during these behavioral tests. Here we'll see a small video of what that looks like. So this is a habituation. The mouse is just getting used to its enclosure, and over on the right-hand side, we can literally see the neuron activity of the mouse. Here's a social interaction test, in which we can see a mouse is being exposed to another mouse, which is contained by a wire container. And again, we can see the neuron activity of the mice as they interact. Here's a social novelty test, meaning one of the mice is familiar and the other one is completely new to our test mouse. As expected, this mouse is spending significantly more time around the new participant. Some mice suffering from neurodegenerative disorders do not show that preference for the new mouse, indicative of a working memory disorder. So, after that video is captured, it's then important for us to analyze the data. This calcium imaging neuron activity video is analyzed by a software program, and data is extracted from it. Here we see a representative image of what some of that data may look like, and a mouse that was exposed to cocaine before and after. So we can see some of the data here. We see a wide variety of charts and graphs that illustrate differences in neuron activity. From these charts and graphs, data can be extracted that shows us the true picture of how the circuit mechanisms of neurodegenerative disorders generally function. This is important because it allows us to specifically target areas of the brain where we know neuron activity is most impacted, and it allows us to have more specific therapeutic treatments that engage in the parts of the brain that are specifically impacted by different neurodegenerative disorders. Using a mouse model is an extremely convenient way to be able to be somewhat analogous to human neurodegenerative disorders, as mice and humans are rather similar in their physiological makeup. So to that end, the actual application and goals of our lab are as follows. So we explore the circuit mechanisms of various neurodegenerative disorders. We also have explored circuit mechanisms of other things, of non-degenerative disorders, such as autism in the past. So that is a really important and crucial way of elucidating information about these disorders and these diseases so that they're better able to be treated in the future. Speaking of which, this year we actually got engaged with a partnership with Johns Hopkins University, which is where Dr. Lee did her postdoctoral studies, to test their new drug. This new drug treats Alzheimer's disease, and they sent us a variety of mice for us to do behavioral tests and the grin implantation and miniscope implantation on. We video them while they're doing these behavioral tests and make sure to record their neuronal activity. And it's a blind study, meaning we are unaware which mice have been administered the treatment for the Alzheimer's disease and which mice have merely been given a placebo. To that end, we are more able to be objective and neutral in our analysis of their data and information. This is crucial because it allows us to be the objective third party, and it allows us to see the impacts of these new drugs as they are treated. To that end, my project is currently the radial arm maze. The radial arm maze is an 8-arm maze that tests for working memory. We put a little food pellet in each of the ends of the maze. So there, and there, and there, and there. So when we put the mouse in a perfectly working mouse, we expect them to visit each arm in succession, going from one to the next, to the next, to the next, to the next, eating the food pellet and not making any mistakes. When they make mistakes, it's referred to as an error in working memory. And this is especially pronounced, obviously, in mice suffering from neurodegenerative disorders. So we can log the number of reference memory errors and working memory errors that they make, using the two groups as a comparison. Here, the black is a normal control, or the black, is a mouse suffering from a neurodegenerative disorder, while the white is a mouse suffering from nothing, a placebo mouse. Not a placebo, a control mouse, my bad. So there we can see the impacts of this. My project is actually a rather time-consuming one because each batch of mice takes about a month to get through the radial arm experiment. And we have at least 3 batches this year, meaning I'll spend quite a large amount of time on this. However, it's a great test of working memory, and it's indicative of a wide variety of factors and edges. So that's what the lab does. Basically, we explore neurodegenerative disorders by looking at neuron activity. We're using that information then in a wide variety of tests, including object recognition tests, social behavior tests, and the radial arm maze test. From that data, we extract important and crucial information about the insights on the functionings of neurodegenerative disorders. And then furthermore, we're able to use that information, as Johns Hopkins has done, in creating new treatments for these diseases. Thank you for listening to my presentation. Please direct any questions to me.